11.2.3 Plasmolysis. The behavior of the plant cell or tissue with regard to water movement depends on the surrounding solution. If the external solution balances the osmotic pressure of the cytoplasm, it is said to be isotonic. If the external solution is more dilute than the cytoplasm, it is hypotonic, and if the external solution is more concentrated, it is hypertonic. Cells swell in hypotonic solution and shrink in hypertonic ones. Plasmolysis occurs when water moves out of the cell and the cell membrane of a plant cell. Plasmolysis occurs when water moves out the cell and the cell membrane of the plant cell shrink away from its cell wall. This occurs when the cell or tissue is placed in a solution that is hypertonic, has more solid. To the protoplasm, water moves out. It is first lost from the cytoplasm and then from vacuole. The water, when drawn out of the cell through diffusion into the extracellular outside cell fluid, causes the protoplast to shrink away from the walls. The cells is said to be plasmolyzed. The movement of water occurred across the membrane, moving from an area of high water potential the cell to an area of lower water potential outside the cell. Figure 11.5. What occupies the space between the cell wall and the shrunken protoplast in the plasmolyzed cell? When the cell or tissue is placed in an isotonic solution, there is no net flow of water toward the inside or outside. If the external solution balance the osmotic pressure of the cytoplasm, it is said to be isotonic. When water flow into the cell and out of the cell and are in equilibrium, the cell are said to be placid. The process of plasmolysis is usually reversible when the cells are placed in a hypotonic solution, higher water potential or dilute solution as compared to the cytoplasm. Water diffuses into the cell, causing the cytoplasm to build up a pressure against the cell wall. That is called turgor pressure. The pressure exerted by the protoplast due to entry of water against the rigid walls is called pressure potential psi p. Because of the rigidity of the cell wall, the cell does not rupture. This turgor pressure is ultimately responsible for enlargement and extension growth of cells. What would be the psi p of a flaccid cell? Which organism other than plant possesses cell wall? 11.2.4 Imbibition. Imbibition is a special type of diffusion when water is absorbed by solid collide, causing them to enormously increase in volume. The classical example of imbibition are absorption of water by seed and dry wood. The pressure that is produced by the swelling of wood had been used by prehistoric men to split rock and builders. If it were not for the pressure due to imbibition, seedlings would not have been able to emerge out of the soil into the open. They probably would not have been able to stabilize. Imbibition is also diffusion, since water movement is along a concentration gradient. The seed and other such materials have almost no water, hence they absorb water easily. Water potential gradient between the absorbent and the liquid imbibed is essential for imbibition. In addition, for any substance to imbibe any liquid, affinity between the absorbent and the liquid is also a prerequisite. 11.3 Long distance transport of water. At some earlier stage, you might have carried out an experiment where you had placed a twig bearing white flowers in colored white and had watched in turn color on an examining of cut in of the twig. After a few hours, you had noted the region through which the colored water moved. That experiment very easily demonstrated that the path of water movement is through the vascular bundles, more specifically the xylem. Now we have to go further and try and understand the mechanism of movement of water and other substances of a plant. Long distance transport of substances within the plant cannot be a diffusion alone. Diffusion is a slow process. It can account for only short distance movement of molecules. For example, the movement of a molecule across a typical plant cell about 50 micrometer takes approximately 2. Can you calculate how many years it would take for the movement of molecule over a distance of 1 m within a plant by diffusion alone? In large and complex organisms, often substances have to be moved across very large distances. Sometimes the site of production or absorption and site of storage are too far from each other. Diffusion or active transport would not suffice. Special long distance transport system become necessary so as to move substances across long distances and at a much faster rate. Water and minerals and food are generally moved a mass of bulk flow system. Mass flow is a movement of substances in bulk on in masses from one point to another as a result of pressure differences between the 2 points. It is a characteristic of mass flow that substances, whether in solution or in suspension, are swept along the same pace as in a flowing river. This is unlike diffusion where different substances move independently. Depending on their concentration gradient, bulk flow can be achieved either through a positive hydrostatic pressure gradient, example a garden hose, or a negative hydrostatic pressure gradient, example suction through a straw. The bulk movement of substances through the conducting of vascular tissue of plants is called translocation. Do you remember studying cross section of root, stem and leaf of higher plant and studying of vascular system? The higher plant have highly specialized vascular tissue, xylem and phloem. Xylem is associated with translocation of mainly water, mineral salts, some organic, nitrogen and hormones from root to the aerial part of the plant. The phloem translocates a variety of organic and inorganic solute, mainly from a leaf to other part of the plant. 11.3.1 How to plant absorb water. We know that the root absorbs most of the water that goes into plants. Obviously, that is why we apply water to the soil and not on the leaves. The responsibility of absorption of water and minerals is more specifically the function of the root hairs that are present in millions at the tip of the root. Root hairs are thin, bold, slender extension of root epidermal cells that greatly increase the surface area of absorption. Water is absorbed along with mineral solute by the root hairs purely by diffusion. Once water is absorbed by root hairs, it can move deeper into root layer by two distinct pathways: apoplast pathway, symploid pathway. Apoplast is the system of adjacent cell walls that is continuous throughout the plant except at the Casparian strip of the endodermis in the root. Figure 11.6. The apoplast movement of water occur exclusively through the intracellular spaces and the walls of the cell. Movement through the apoplast does not involve crossing the cell membrane. This movement is dependent on the gradient. The apoplast does not provide any barrier to water movement, and water movement is through mass flow as water evaporates in the intracellular spaces or the atmosphere. Tension developed is a continuous stream of water in the apoplast. Hence, mass flow of water occurs due to the adhesive and cohesive properties of water. The symplastic system is the system of interconnected protoplast. Neck boring cells are connected through cytoplasmic strands that extend through plasmodesmata. During symplastic movement, the water travels through the cell. Their cytoplasm intercellular movement is through the plasmodesmata. Water has to enter the cell through the cell membrane, hence the movement is relatively slower. Movement is again down a potential gradient. Symplastic movement may be aided by cytoplasmic streaming. You may have observed cytoplasmic streaming in cell of hydrilla leaf. The movement of chloroplast due to streaming is easily visible. Most of the water flow in the root occurs via the apoplast. The cortical cells are loosely packed and hence offer no resistance to water movement. However, the inner boundary of the cortex, the epidermis, is impervious to water because of a band of suberized matrix called a Casparian strip. Water molecules are unable to penetrate the layer, so they are directed to wall regions that are not suberized into the cells proper through the membrane. The water then moves through the symplast and again crosses a membrane to reach the cell of the xylem. The movement of water through the root layer is ultimately symplastic in the endodermis. This is the only way water and other solid can enter the vascular cylinder. Once inside the xylem, water is again free to move between cells as well as through them. In young root, water enters directly into the xylem muscle and or tracheids. These are non-living conduits and so are part of the apoplast. The path of water and mineral ion into the root vascular system is summarized in figure 11.7. Some plants have additional structure associated with them that help in water and minerals absorption. A mycorrhiza is a symbiotic association of fungus with a root system. The fungus filament form a network around the young root or they penetrate the root cell. The hyphae have a very large surface area that absorb mineral ion and water from the soil from a much larger volume of soil that perhaps a root cannot do. The fungus provides mineral and water to the root. In turn, our root provides sugar and ink-containing compound to the mycorrhizae. Some plants have an oblique association with the mycorrhizae. For example, pinus seed cannot germinate and stabilize without the presence of mycorrhizae.